excited. We've got Gözde Çilingir joining us. She is a nature conservationist, geneticist, and conservation advocate. So as a postdoctoral fellow at the University of Zurich in Switzerland, she works to bridge the gap between scientific research and real-world application for genetic conservation of Aldabra giant tortoises native to the Seychelles. And I think this is so cool. We work with the Seychelles Island Foundation, and we do live events for classrooms where we visit the Vallée de Mai and Aldabra as well, and connect with scientists working on the islands. They've just got a great new communication system on Aldabra, and it's going to allow us to do some really cool live events in the coming months. I'm excited about. So she has led and contributed to several turtle conservation outreach programs around the world, including Singapore, Thailand, Myanmar, and Turkey. So let's bring her in live with us right now. Hello, Gözde, how are we doing?
Hello, Joe. I'm doing fine, thank you. And how are you?
Oh, great. I mean, it's just been a wild weekend. The time is flying by. I can't believe we're in our last kind of run of events for the weekend. So many great stories. I'm so excited to have you joining us. I'm going to take a back seat. I'm going to throw myself backstage, and I want you to take over a bit and take us into your world.
All right, thank you. So I'm going to share my presentation. Just a second. Okay. All right, I hope you can see me and hear me. So I will be mainly talking about genomic aspects in the conservation of threatened turtles. Before going into my research, the details of my research, I would like to start with my background. I was born and raised in Istanbul, Turkey, and I got my bachelor's degree from Istanbul Technical University at the Department of Molecular Biology and Genetics. I have basically a solid biotechnology background, but after several years, I decided to go on for ecology and nature conservation, and I did my master's in Middle East Technical University on phylogeny and phylogeography of brown bears in Turkey. Then I moved to Singapore to work on critically endangered turtles, freshwater turtle, endemic to Myanmar and Cambodia. So today I will be talking about a part of my PhD research and my current postdoctoral research, and I also would like to talk about my life as a conservation geneticist. When I tell people that I'm a conservation geneticist, sometimes they get puzzled. They ask if I clone some rare turtles to increase their numbers. But actually what I do is I try to understand the remnants of genetic variation in the threatened populations or species, and I try to find ways to conserve that variation, hoping that their adaptability for changing environment will go hand in hand with that retained genetic variation. There is also another thing that I'd like to mention before diving in what I have been doing for the last 8-9 years is the distinction between the science of nature conservation and conservation biology. So basically nature conservation is a multidisciplinary field, which includes social sciences and natural sciences, and conservation biology is actually a subset of it. And conservation genetics is a subset of conservation biology. So the case studies that I'm going to mention here is basically a component of a bigger conservation projects that have been going on for several years. Why turtles? So of all the known turtle species in the world, I mean, half of them are threatened with extinction. And here on the map, you see the number of threatened species on a given region. And as you can see, Southeast Asia is one of the hotspots of threatened turtle species. And for my PhD, I was working on Burmese roof turtle, which is endemic to Myanmar, one of the rarest turtles left in the world. And although they were widely distributed around Myanmar once, unfortunately, they got into a point that they were thought as extinct. And finally, I mean, there are several populations left. They were rediscovered, and it was known that there were several populations left, but currently we know that there is only one population left in the world from this turtle species, and it is located in the northwestern side of Myanmar, in rural Myanmar. And this population was kind of ecologically extinct because there were very little number of breeding females that was thought as less than 10, depending on the field observations, but the number of breeding males was unknown prior to my PhD. And once the population was spotted, Wildlife Conservation Society and Turtle Survival Alliance initiated in situ and ex situ conservation together. Basically, the beaches that were used by the females for egg laying was under protection. And then turtle conservation team there collected those eggs in the wild, and they transferred them into secure places, reared those hatchlings there. And by doing this for several years, they ended up with having hundreds of individuals in captivity from this very rare turtle species. So my PhD came into play at this point, because the ultimate goal of this conservation project was to have self-sustaining turtle populations in their natural habitats. But if you have hundreds of individuals from such a rare species, would you introduce all of them together in the wild? Probably not, right? So my project was aimed to understand the current genetic variation and the relationship between these individuals, so that I could manage these genetic variation and pick individuals to be reintroduced into the wild. And also, because we had no idea about the numbers of breeding females and males in the wild, I would do parentage analysis to the individuals that we have in captivity, like as we do in humans, and try to estimate the numbers of the wild breeders. To do that, I went to Myanmar several times and collected tissue samples, extracted DNA, and then came the question, which methodology to use. So genome sequencing, as you may have heard, is becoming more and more cheap and feasible, but still, sequencing the whole genome of many individuals at the same time is still too costly and too laborious and computationally too costly. So for the conservation projects that you have to decide quite quickly, this type of whole genome approaches would not be suitable. So I had to decide quick, and I had to find a way to make this budget friendly and feasible for these individuals, for this species, where we had no genetic tools beforehand. And that's why instead of looking at the whole genomes, I basically looked at several parts of the genomes of these individuals and decided which individuals to go into wild. And by doing that, we actually reintroduced these several individuals that I picked into several stretches of the Chinwind River that the species was naturally found at least 2 times at the initial step, as an initial step. But after reintroduction, there was another point, like, what if the worst case scenario happens? What if the reintroductions wouldn't work? So we needed to have a genetic stock of the species, which could be contained in Myanmar, that could be located in Myanmar, as well as outside of Myanmar, just in case. So in order to have such colonies, which are named assurance and satellite colonies, I came up with several lists of individuals that were picked by genomic approaches, and 25 individuals picked, as I explained, were sent to Singapore Zoo. So currently there are 25 individuals in Singapore Zoo that were transferred from Myanmar, and these individuals actually stand as a genetic stock for the entire species. Okay, this was the conservation of the genetic diversity of a species, that aspect. But how about the human perspective of this story? So these individuals, this species is at the brink of extinction because of human behavior, basically, like pollution, habitat destruction, or overexploitation. So the idea of our secondary project in that area was to visit the villages alongside the Chinwind River, where these individuals have already been introduced. And by interviewing the villagers, they try to understand the attitudes, local people's attitudes towards turtle conservation in the area. To do that, we visited at least 30 villages in one season with my local collaborators there. And we asked several questions to the villagers. Because there was very restricted land connection between the villages, we were taking a boat to visit all those villages, and we were basically being guests of the villagers there. So the idea was to show some pictures of animals, because these animals were so rare. Most of the time, people couldn't recognize them, or it was important for us to understand if local people could distinguish them from the other turtle species in that river. And if they could, we also started asking questions if they could understand or distinguish the nests or eggs of the species, if they have ever seen such nests, if they have heard some stories about the species, if they have heard about our conservation programs in the region, or if these programs restrict their daily lives, and so on. So it took us more than 1.5 years to come up with all those questions and do the field work and come up with conclusions. And at the very end, the idea was to finish the surveys and try to reach as many people as possible to explain what we have been doing. And as a result of this project, we realized that because local people's daily lives weren't that much restricted because of ongoing conservation work, they were willing to contribute to the ongoing projects, or there's no problem of contributing to the project. And also, because these species this was rare for so long time, many people had no direct contact with the species, but they kind of consider the species as a heritage from their grandparents, because they have heard stories from their grandmothers or grandfathers. That was an interesting aspect, interesting way to realize that. And after each visit, we also try to summarize what they have been doing. And this also gave me the opportunity to visit too many sites in Myanmar, get in contact with local people, understand their needs, understand the local habitat of the species, and as well as meet too many nature conservationists and conservation scientists in Myanmar. And after all these projects, then comes the other question, like, there are ecologists in Myanmar, there are nature conservationists in Myanmar, but why haven't they been doing this work that I have been doing in Myanmar by themselves? And the answer for that was lack of facilities to provide some facilities that were lacking there, and also to bring the know-how that we have built up, that we have come up with during my PhD. We initiated a project with my local collaborators in Myanmar to basically have a seminar for the nature conservationists in Myanmar to come together and discuss what we have doing in the country, and also to come up with do-it-yourself molecular laboratory in one of the universities, and give students there who have been working on ecology and nature conservation for their thesis to explain the equipments and how can they incorporate those equipment into their thesis, and also have some volunteer students from the university and bring them to our field sites. And the field site, they could meet secondary school students and bring a portable microscope with them, show them how to distinguish plant and animal cells under a microscope, and where DNA is found in those cells, and what does DNA or genetic diversity mean, and what we have been doing in there, different turtle species in Myanmar, and turtle conservation projects going on in the region. By doing that, we actually managed to diffuse the scientific knowledge in different educational bodies through Myanmar. And this was actually the first time that I got involved in National Geographic community. And we still are in contact with my collaborators or university students there. But unfortunately, because of political situation in Myanmar right now, they are distressed. And if you are interested in to learn more about what's going on in the country, you can actually search for hashtag what's happening in Myanmar. Currently, as I said, and Joe also introduced me as, I'm a postdoctoral research at University of Zurich. And my topic here is on the population and conservation genomics of Aldabra giant tortoises. Aldabra giant tortoises, together with Galapagos giant tortoises, are one of the 2 giant tortoise species left in the world, and their natural distribution restricted in Aldabra atoll. Although there are many other several giant tortoise species in Western Indian Ocean islands, they're all extinct. So Aldabra giant tortoises are the only remaining ones in the region. And they have been successfully used in several rewilding programs. By rewilding programs, I mean these individuals, these animals were actually sent to other habitats, the degraded habitats, and they could actually substitute their extinct counter species. So they basically substitute their ecological roles and then help resurrect degraded ecosystems in the region. So the idea was previous work on the genetics of Aldabra giant tortoises were done with a handful of molecular markers, and they were inconclusive. And my work basically is about using a different approach, which looks at the whole genome for many individuals and to have a more in-depth idea about the genetic variation of the individuals. We need such information, again, to understand how genetic variation is partitioned in natural habitats and how we can actually manage this variation when it comes to rewilding programs or when it comes to the conservation of the species. This information is important because during rewilding, we basically introduced several individuals from a large population, right? So managing the variation so that the introduced population would represent what is left in the wild is quite important. In addition to that, I recently went to Mauritius for my fieldwork because when we introduce several individuals in the wild, we kind of assume that these individuals will mate randomly, so the genetic variation will be retained. But in general, this is not the case. And we have no idea about the breeding system of the species. To understand the breeding system, I basically sampled newly born tortoises in Mauritius, and I will basically do some parentage analysis for them and to understand, in one nest, how many fathers were there, how many breeding females mated with how many males, is there any male dominance, and etc., etc. So all the information that we're going to gain from this project will actually help us to more effectively manage future rewilding or future introduction programs. Another program that I'm involved recently, have been working on recently, is to understand the age, estimate the chronological age of Aldabra giant tortoises. This is important because by knowing the age of individuals, we can also model the dynamics of their populations. We could model the dynamics of wild populations and understand how evolutionary mechanisms played a role into their current genetic variation or population structure. Plus, we could also do this same model, same approach, use the same approach to plan for wilding programs. And also, this is important for the welfare of the individuals as well. To know how old they are will also mean that how we should care for them. And also, Aldabra giant tortoises are one of the oldest known land vertebrates, and that would be super cool if you could estimate the age. So one could ask why you could estimate age of these individuals with molecular tools. Cannot you do any other thing to understand this? Well, for tortoises, this is not the case. We have no non-invasive or non-harmful method to estimate the age of the Aldabra giant tortoise. There's no phenotypic cues. And how we can do now is basically by looking at their genomes and several patterns on the genome and correlate this pattern with the known age of some individuals and build an age clock model for the species. And by using this model, we could also estimate the age of the ones in the wild, in their wild populations. So all in all, what I have been trying to do for this threatened turtles is I try to find cheap, feasible, and depending on the situation, rapid way of incorporating genetic and genomic aspects on the real world conservation programs of these turtle species. And in addition to that, I also like and would love to do in the future as well, to bring scientific capacity and know-how to the places that I work, so that our circle of nature conservation group would enlarge, and then these genomic tools that I came up with could be used by anybody who would really like to use, especially for the conservation of these populations and species. But of course, this is not, I mean, all these case studies that I mentioned could not be done without the help of my collaborators and the funders. I am thankful to all of them. And I also would like to thank you for your kind attention.
All right. Well, perfect slide to end off on with our Teenage Mutant Ninja Turtles. I think still popular to this day. I remember growing up and having all those action figures. So that brings me to the first question is turtles. What led you to want to get into this field and start researching turtles and looking at ways to kind of preserve those populations?
That's a great question. So the issue is when I tell people that I spent last 8-9 years of mine to conserve some turtle species, people tend to assume that I have some previous story behind, like, from my childhood or any other special interest in reptiles or turtles before. But in fact, I didn't have any. I worked with brown bears, honeybees, deer, several other different animal groups before. And when I started my PhD, I just wanted to use my molecular background to nature conservation project. I didn't have any particular animal group in mind, but my PhD supervisor kind of listed the projects that he had at that time. And I basically picked Myanmar instead of turtles. And then it started. Because they have fascinating life histories. As a geneticist, I am super interested in turtles right now. But at the very beginning, I basically picked Myanmar.
Hey, and there's nothing wrong with that, right? There's no right or wrong way to get into science.
Sure.
You don't know where your career is going to take you. And kind of being open to trying new things, being curious, and look where it's led you. You've visited some amazing places. You got to work with some iconic species like the Aldabra giant tortoises. So not too bad of a choice.
Exactly.
All right. Well, what I loved about the projects that you shared with us, particularly the first project, is you attacked it from every angle. So not just the science side of things, then you went out and brought the community in. And then, you work to bring some of the local conservationists into the project as well. So this approach is obviously been successful. Is this something that you've been able to share with other research projects and other organizations?
So the issue is, I think one of the downsides of being a geneticist and doing nature conservation is sometimes you tend not to go to field that often. If somebody brings your samples to you, you could basically do your work in the lab and in front of a computer, and then you could publish or share your results. It's okay. I was lucky because my collaborators in Myanmar there were so willing to host me in Myanmar. And every time I go to Myanmar, I tend to get in contact with too many people and tend to understand the roots of the problem there in terms of nature conservation. So we came up with more questions, more possibilities. So we worked really tight, and our main goal was to just solve those problems as soon as possible, and incorporate science-driven proofs or information as much as we can. And when I mentioned turtle conservation team, I mean, nature conservationists, scientists or field staff, NGOs, former government of Myanmar, too many people with different backgrounds. And from Singapore side, I also had many people like social scientists interested in this project. And we all know that if we want to solve a complex problem, we have to come up with different angles. We have to come together and work together. That was our main drive.
Yeah, absolutely. We're in a situation right now where we're losing species. We're losing them fast. In some cases, we're losing them before we even know they existed. But what you're doing is so important, is adding this piece to conservation where we look at the populations we have left of some species, which are actually really small in many cases, and making sure that the right individuals are being used to further these populations. And so I wonder what advice you would have for somebody who's maybe starting their science career and maybe hasn't looked at conservation from this angle before. What advice would you give them to approach this field?
So I would advise for a person who would be interested in conservation genetics or genomics in general, my main advice would be, for them to think as practical as possible. Because as I said, theoretically, everything could look fine on paper. You could come up with a list of individuals and you can say, yes, this individual could go this way. But sometimes things don't work like that. My main advice would be, it also depends on the projects. There is no I don't have a solid advice, but I would say, not looking from a single perspective into of any conservation related problem, because usually the problem is not that easy. If the problem is not easy, then we cannot solve it by looking with only one single direction. And genetics is a small component of many conservation studies. That is why it is hard to do applied conservation genetics. It tends to stay on paper. It tends to stay theoretical. And because it's expensive, too costly in many ways, it is really hard to show the applicable part of it. So my advice would be meet lots of people, have lots of local collaborators, and be interested in other aspects of the conservation problem, so that people could also think in more practical ways, or how to translate their scientific findings into a solution in the real world.
Awesome. Great advice. And that kind of leads to our final question here. We have another question here via YouTube. And this person is wondering, are you still in touch with that team in Myanmar? How are things going with that project?
Yeah, I am. Well, regarding the project, I can say that after the reintroduction, let's say before the reintroduction, we couldn't get fertilized eggs for several breeding seasons. But after reintroduction, we started to get fertilized eggs, and we kind of speculated that this could be because of the effects of reintroduction. Effects of males enter the gene pool of the population. So after several years, we were really hopeful about these reintroductions. But currently, I can tell the captive conservation of the populations have been going stable. But I am not sure if they're planning several other new reintroductions in the region, given the political situation right now.
Yeah, absolutely. And that's another aspect to think about, is you may have those kind of stable genetic populations, but then is it the right time, the right conditions to be furthering the project, right? So another thing to consider, and why it's so important to have those stable populations in places like the Singapore Zoo. Yeah. Awesome. Well, Gözde, thank you so much for spending some time with us today, for joining year 2 of the Women Blaze Trails Festival. I'm going to share your handles here on social media. There they are. If people would like to follow along, learn more about the work that you are doing. And I'm so excited that you're doing work. I saw Luke's name there in the Aldabra research. So I'm so excited that you're doing work with that amazing team. And who knows, maybe we'll be able to connect on an event somewhere down the line, maybe even from the field.
Maybe, hopefully. Thank you very much for the invitation, Joe. It was so nice for me to be here. I was so excited to contribute to this fantastic festival, and hope to meet you somewhere else.
Awesome. Well, I look forward to that. Have a great rest of the weekend, and we'll talk soon.
Thank you. Bye.
See ya. Thank you.